Hi! In this video, the introduction and demonstration of our work in paper ID 7254, a 75.6 megabytes base pairs per second FPGA accelerator for FM index-based paired end short read mapping will be presented. First, here's an introduction.
With DNA, we can analyze gene expression, which enables precision medicine, genetic testing, and species determination. So how do we get the DNA data? Sequencing. To sequence the first human genome, it took 20 years and over $3 billion. With the advance of technology, the next generation sequencing can sequence the whole human genome in a single run, and the cost can be reduced to $1,000. But there are some new challenges. The next generation sequencing first breaks the long string of DNA into tens of millions of smaller pieces. The small pieces of DNA are also called short reads. These short reads have to be assembled to rebuild the whole human genome by special algorithms. However, the NGS data analysis tools today are still not efficient enough to solve this problem. Even with high-end GPU, it still takes over 3 days to complete the whole human genome data analysis process.
In NGS data analysis, there are 4 main steps, which are short read mapping, haplotype calling, variant calling, and genotyping. In short read mapping, all the short reads from the DNA sample are aligned to the reference DNA to reconstruct the original genome. From haplotype calling to variant calling, at first, the mapped short reads are connected by the overlapped parts to form haplotypes. Then, the differences between the DNA sample and the reference DNA sequences are identified, which are called variants. Lastly, in genotyping, the most likely genotypes are determined by calculating the likelihoods of all possible genotypes. In order to accelerate the time-consuming NGS data analysis, which is the bottleneck of the workflow, we implemented an FPGA accelerator for short read mapping. 4 techniques are proposed to enhance the throughput, which include 1. OCC-BWT data interleaving, 2. Two-stage LF mapping, 3. Repetitive candidates filtering, and 4. Early termination. For OCC-BWT data interleaving, according to our analysis, the occurrence table and Burrows-Wheeler transform data are required at the same time. Therefore, they can be reallocated in an interleaved manner so that they can be accessed by just one DRAM access, reducing the latency by 51.8%. The original LF mapping is performed to find all the possible locations of short reads. The required iterations are equal to the length of the subsequence of the short read, which is called seed. To reduce the latency, a lookup table is pre-built to record the meta-results of LF mapping, which can be used as the initial values of the necessary parameters in the second stage of LF mapping. Thereby, 60% of the operations can be skipped, reducing the latency by 62.2%. For repetitive candidates filtering, the candidate locations that are already identified can be filtered out in advance to skip the compute-intensive dynamic programming for calculating the similarity score between the reference subsequence and the short read. This reduces 10.9% of the latency. For early termination, since most of the candidate sequences are exactly matched for one short read, the procedure can then be terminated early when any of them is found. This reduces 54.3% of the time and latency. Before we start, let us introduce the demonstration system. For the hardware setup, the demonstration system integrates 2 AMD Xilinx Alveo U250 FPGA boards. The proposed high-throughput short-read mapping accelerator is implemented on one board, and the others are implemented on the other one. The PCI Express interface is used to connect the 2 FPGA boards and the host PC. A graphical user interface is designed for selecting the reference genome, tests NGS data, and output folder. It also shows the progress and the results of the task. Here is our demonstration. Before the analysis process begins, the reference genome is loaded. Due to the time limitation, we'll be using the whole exome sequencing data as a test dataset, which is the data among the more informative regions of the DNA sample. The proposed short-read mapping accelerator is activated by pressing the Start button. The processing status is shown on the rightmost column. After that, the system proceeds to haplotype calling, variant calling, and genotyping on the other FPGA. The entire data analysis is finished in 10 minutes. The results of the variant discovery are then stored in a standard .vcf file. This system also shows the results by a visualized genome browser. The outputs of the demonstration system can be used for further diagnosis and interpretation by researchers and doctors. The proposed FPGA accelerator for short-read mapping achieves the highest mapping accuracy of 99.3% and 1.7 to 18.6 times higher throughput compared to the state-of-the-art FPGA-based solutions. This concludes our demo. Thanks for watching.